At Arizona State University, researchers have made a significant breakthrough in gene regulation within living organisms. Their recent study, published in the journal Nucleic Acids Research, focuses on the essential role of specific RNA fragments in the small, transparent roundworm Caenorhabditis elegans, C. elegans. This groundbreaking study provides a detailed map of the 3' UTR regions of RNA in C. elegans. The 3' UTRs, or untranslated regions, are critical segments of RNA that play a role in gene regulation. With this new map, scientists now have a valuable tool to study how DNA genes are activated and deactivated after being transcribed into RNA. Using the data from this study, researchers can improve their predictions on how small RNA molecules, miRNAs, interact with genes to control their activity. They also explored crucial regions of the 3' UTRs that help in processing and regulating RNA molecules. This detailed exploration offers new insights into the intricate processes of gene behavior. This monumental work represents a culmination of 20 years of hard work. We finally have the complete picture of how genes are formed in higher organisms, says Mangone, the corresponding author of the study. With this complete dataset, we can now pinpoint and study all the regulatory and processing elements within these gene sections. Genes are segments of DNA that contain the blueprints for the astonishing diversity of life on Earth. However, part of the secret to this versatility lies not in the genes themselves, but in how their effects are finely tuned. Genes provide the instructions for making proteins, which play essential roles in building and repairing cells and tissues, speeding up chemical reactions, and defending the body against pathogens. During the process of protein production, DNA is first copied into RNA, which acts as a bridge between the DNA template and the resulting proteins. Although our DNA genome is fixed from birth, RNA provides the body with enormous flexibility by regulating how genes are expressed. Once genetic instructions are transcribed from DNA into messenger RNA, mRNA, specialized segments of the mRNA, the 3' UTRs, regulate how proteins are produced. These 3' UTRs are located at the end of a messenger RNA molecule and control the stability and efficiency of the mRNA. Initially, non-coding RNAs like 3' UTRs were regarded as non-essential genetic fragments because they do not code for proteins. However, recent research reveals that they are crucial for modifying gene behavior and influencing mRNA stability, localization, and translation efficiency. These regulatory elements often contain binding sites for other protein-regulating elements, including microRNAs and RNA-binding proteins. Despite their importance, scientists previously knew little about 3' UTRs. This new study addresses that gap by mapping out 3' UTRs for nearly all genes in C. elegans, providing the most complete map of its kind for any animal. C. elegans is a small, transparent nematode extensively studied in biological research. Its simplicity, short life cycle, and well-mapped genetic structure make it invaluable for studying gene function, development, and disease processes. Its transparent body allows researchers to observe cellular processes in real time, and its genetic composition enables precise gene manipulation. The study discovered that switching between different 3' UTRs is less common in C. elegans than previously thought, challenging earlier beliefs and highlighting the complexity of gene regulation. With this new data, scientists have updated predictions on how microRNAs interact with genes. The insights gained from this study have far-reaching implications for human health. Problems with gene control can lead to diseases like cancer, diabetes, and neurological disorders. By providing a detailed map of 3' UTRs and their regulatory elements, this research offers new insights that could lead to better treatments and therapies. The new dataset produced by the study will be a key resource for scientists studying genetics and human health. The ASU team plans to continue their research to further explore how these regulatory elements work and their critical influence on gene control.